Okay. All right. Well, welcome everybody. It's my great pleasure to start the next session with our first speaker this next session is actually translational. And so the first speaker will be Neil Hanchard, who's currently at the NIH. He recently moved there from Baylor College of Medicine. And he'll be speaking on translational research for childhood complex disease. Neil received his medical degree from the University of West Indies, his Doctor of Philosophy from Oxford University, and then he completed his medical training in the US at the Mayo Clinic and the Baylor College of Medicine. He uses a combination of quantitative and integrative biological and genomics methods to understand complex pediatric disease, and particularly in populations of diverse ancestries, and emphasizing this kind of interplay between populations and genetic susceptibility, an area that is very close to my heart. And Neil, I will now hand over the podium to you, and I'm looking forward to your talk.
I guess I should ask, can you hear me? Wait, I'm on mute. That's what I got to do. Okay, can you hear me?
Yes.
Okay, and my presentation's available. Okay, great. Well, first I want to thank the organizers for the invitation, the opportunity to sort of share your research with the communities and groups that have most empowered it is humbling and really an honor. So I've asked to talk about sort of a translational approach to childhood disorders, and we accomplish that mostly using genomics. And so what I thought I would start with is giving an overview of our process, and then maybe speak specifically about one of the specific disorders. So as a physician scientist, I usually start these things by thinking about a particular disease or a clinical phenotype, and then thinking about the epidemiology and the demography of that to be able to inform an appropriate genetic model. So you might end up with very rare diseases that fit the Mendelian mode, for which there are very few genes and very high effects, or you could be at the other end of the spectrum. And so once we've sort of thought about a genetic model to use, then we sort of recruit cohorts accordingly, and then we can apply sort of the modern genomic technologies. And that really forms a nidus for discovery. And that's the starting point to then funnel our way through sort of mechanism and using models to understand how we could best sort of have a targeted intervention, with the hope that ultimately we might be able to close the loop in going from bedside to bench and back. So we're doing this on a number of different fronts. You can see the diseases that we're interested in here, and all of these have a very heightened prevalence in populations of African ancestry, which makes this more prescient. So what I thought I would do today is to talk you through some of the work we've done on childhood malnutrition using this kind of genomics approach. So severe acute childhood malnutrition is still a major global health issue, with a very high prevalence worldwide, and particularly on the continent. And the major problem, of course, is that it's associated with a very high mortality, both indirectly and directly, particularly in children under the age of 5. Now, you can say, well, you know, the way that you treat severe malnutrition is sort of just have food security. But we've known that for a while, and still there are a number of sort of geopolitical and climate issues that often intervene to keep that from being a reality and make it still a very prescient disorder to consider. So what's less well considered sometimes with severe malnutrition is that there are kind of two prevalent forms. The first is the sort of edematous form, which goes by the names of kwashiorkor or marasmic kwashiorkor, and is characterized by sort of multi-system involvement, including the hair, the skin, and then pretty much every other organ system you could imagine. And is characterized by swelling or edema of the body. And this form is classically much more difficult to treat and has a higher mortality when compared to the non-edematous form, which is also known as marasmus, which is predominantly characterized by wasting. And although we've known these two forms for some time, it's actually not 100% clear why you get one and not the other. And some of the proof of principle of that is that if you look at the treatments, it's the same for both forms, despite them having different outcomes and different pathophysiologies. And this is not a new disease. So there are those who have sort of pointed to references to the Israelites wandering around in the desert and comments about their feet not becoming swollen as some of the earliest references to kwashiorkor. But the first official mention in the medical literature was in 1933 from Cicely Williams, who was a Jamaican pediatrician who was working in Ghana, in Accra at the time. And since that original description, there have been a number of studies, and here this is just the publications in PubMed that list kwashiorkor. And those studies have predominantly focused on biochemical, physiological, and nutritional differences between the two. And so there's this very large catalog of differences that you can observe between the two groups in their physiology. But despite all these studies, it's still actually not known that there's any single clear definitive etiology. But what is known is that there's no consistent difference in the diet or infectious exposure. And so that has shifted a lot of the focus back to more innate issues, which includes genetics. And so we have taken on thinking about edematous malnutrition or kwashiorkor as a complex multifactorial disorder. And so one of the physiologic metabolism defects that's been observed, that's sort of coming to the fore as maybe most relevant to the disorder, is one, carbon metabolism, specifically the conversion from methionine to homocysteine and back, which is responsible for 80 to 90% of the methyl groups that are produced, including the methylation of DNA, which you see here. And stable isotope studies that were done in Jamaica many years ago demonstrated that when acutely ill, the children who have the edematous form have a much slower turnover in this cycle than those in the non-edematous form. And when recovered, however, there's no difference between the two groups. So we thought that this was a good place to start in terms of thinking about our genomics approach. And that's because DNA methylation is mitotically heritable. By that I mean that when the DNA is replicated, the new strand has to have the same methylation marks placed on it. And so you could imagine that in cells that have a high mitotic rate and they're turning over at some rate, that you could find that with each sort of generation of cell proliferation, this sort of limiting availability of methyl groups and substrates might mean that you could actually eventually see differences in DNA methylation in a bulk way between the two groups, and that might have an insight into the underlying pathophysiology. And so we decided to study this using two cohorts from Jamaica and Malawi. And these were all children, all aged between sort of 0 and 3 years, who had buccal cells done. And those buccal cells were taken in and around the time of their original diagnosis. So this is a cute group, we refer to them as DC. And the numbers are actually relatively evenly split between Jamaica and Malawi. We're also fortunate to have a group of adults from Jamaica who had survived childhood malnutrition and are now adults, and they're healthy otherwise. And so all these individuals were typed on the Illumina 450 carrier or the EPIC array. And so we're looking at just under 500,000 CpG methylation sites across the genome. And so the first thing we did was we just asked, do we see evidence of differences in methylation? So of those sites where we could see, strongly, significantly differential methylation, almost all of those sites at a Bonferroni corrected significance level, almost all of those sites are hypomethylated in edematous relative to non-edematous, which are the red dots. We also saw that this effect was both similar in magnitude and direction in Jamaica and Malawi, and that the CpG loci that were involved are spread all throughout the genome, which you see represented here on the outer ring. And the black dots represent the individual loci the inner ring. So to reduce some of the false positives that we sometimes get with sort of a single locus kind of approach, we clustered these associated regions into differentially methylated clusters between the groups. And you can see those here in the red dots in the outer ring. But interestingly, when we looked at the adults and compared their methylation status, we see no difference between edematous and non-edematous. So this is strongly in keeping with what the stable isotope biochemical studies had shown us, which is that these primary differences are occurring in that acute nutritional event. And then when they get better, there's no longer those differences. So we wanted to give this some additional context. So we looked at where these differentially methylated sites were. And you can see that relative to background, we see an enrichment over the gene body. So the relationship between sort of DNA methylation and gene expression is well understood at promoter regions to be sort of inversely correlated. And so we had some RNA and DNA paired samples from some of the children. So you can see at the promoter region here, there's an inverse correlation, which represented the y-axis, such that you would expect low methylation, high gene expression, and vice versa. But at the gene body, we see both evidence of some negative correlation, but also some positive correlation. And so the overall picture is that although it's hypomethylation, you're seeing this very complex transcriptional response, which is likely to be influencing the complex physiology that we see. So we thought about, well, what genes might be involved in that? And so we put these in the context of gene ontology terms, and you can see again that we're getting some biologic relevance to the underlying biology. So responses to exogenous things like nutrition, things that affect sort of water and solute movement related maybe to edema, and then things that relate to sort of anthropometry and body size related to nutrition. And in fact, if we kind of looked specifically at the individual genes, we could see lots of examples of ones that have been brought out in the literature of severe malnutrition, including sort of fatty liver in sort of rat models, of malnutrition, genes that have been implicated in changes in glycosaminoglycan content in the small bowel of children with kwashiorkor. And then we also saw a number of genes that are related to immunity. And in particular, have been shown to be disrupted in sort of overnutrition states, so obesity or elevated BMI with DNA methylation. And in fact, if we took all of the genes that we observed and sort of went to the GWAS catalog and just asked what, if any, genes are associated with these particular, what disease traits are represented by these genes, we can see again that we see this preponderance of sort of cardiovascular and metabolic disease and things to do with body mass and anthropometry, where you see an accumulation of these kind of genes occurring. So that said, well, maybe we're on the right track biologically, and allowed us to develop this kind of molecular framework as a working framework for going forward. And so that would say, well, in the face of this stress, we have this aberrant 1-carbon metabolism, and then there are downstream disruptions in sort of DNA methylation and other related processes. And that then gives rise to the two phenotypic differences that we see between edematous and non-edematous malnutrition. And the nice thing about this framework is that it does allow for things like the microbiome. So studies have shown where you have changes in the gut microbiome, you get a shift in towards gut bugs that have a very different 1-carbon metabolism, and would also be consistent with a recent mouse study where they fed the mice a sort of diet that's very similar to the one that you see in Malawi and has been associated with malnutrition, where after about 2 weeks, you can see this accumulation of fat, which is these red globules here in this liver biopsy, whereas the same mice given the same diet, but with choline, which is a methyl donor, you actually don't see that accumulation of fat. Now, choline has a lot of other different uses, so this is not exactly like a slam dunk, but it is highly suggestive that we're on the right track. So then we wanted to kind of give this a little bit more sort of shape. And so as a human geneticist, I was really interested in how genetic variation might influence this. And so you could think about the first way, which is that it might influence the DNA methylation as these sort of so-called methylation quantitative trait loci, or QTLs. And actually, we did find evidence of this. So you see that illustrated here, where the degree of methylation in the edematous group is dependent upon the underlying genotype in the acutely ill state, but you don't see that same effect in the non-edematous acute or in the completely recovered group. And this kind of context-dependent DNA methylation QTLs, we found at about a quarter of the sites where we had differential methylation. And so we kind of view these as maybe sort of fine-tuners of the hypomethylation. That is, the kind of things that might make the difference between why you're more severe or less severe, or why you might have more fatty liver or skin or hair changes in one case than the other. But there are other ways that your genetic variation might play a role. And so you could think about, well, what about the genes that are related to directly related to 1-carbon metabolism? And so we did look at this using our full cohort of 700 individuals from Jamaica and Malawi, all genotyped on the H3Africa array. And then we just looked for any locus that has been associated with 1-carbon metabolism in the literature. And then we sort of focused initially on the SNP genotypes that were related to these 105 genes. And so, if we look at those in a Manhattan plot, so again, each dot is a SNP, the height reflects how significant it is. And at a gene level cutoff, and certainly in keeping with the QQ plot and its deviation from expected, we can see there are at least 3 loci that meet this kind of threshold. And of course, then the question is, well, what are these 3 loci? So, all of these loci are going to be related to 1-carbon metabolism in some way. And so PRCL2 is the first one. The exact role of PRCL2 is not precisely known, but as a locus, it's certainly been associated with folate levels and replicated as such. GABBR2 is actually a GABA receptor, but it's the same idea that sort of the upstream region as a locus has been associated with homocysteine levels fairly consistently. And then we have SHMT1, which is actually the enzyme that converts 5-methyl tetrahydrofolate into tetrahydrofolate in the remethylation of methionine. And so at least the top SNPs are sort of all culminating on this area where we're sort of remethylating methionine, which of course is an essential amino acid. So if we look at that in a bit more detail, what we can see is that at GABBR2 and SHMT1, we see there's a higher frequency of the top alleles in the edematous in the non-edematous versus edematous. So this is more of like a protective effect, right? So protecting from edematous forms. And actually the variant in SHMT1 is a strong EQTL, particularly in the thyroid, which is a relevant tissue to malnutrition. And the direction that you see here would be consistent with the sort of resulting enzyme changes that would fit our original hypotheses. At PRCL2, we can see that all of the sort of top SNPs there are all within this block of LD. And it has the same kind of features where it's a sort of a protective type of effect from edematous malnutrition. So we kind of wanted to take our top SNPs and also put them in a different, broader context. And so one thing we thought is that, if you are going to survive severe malnutrition, that might be the kind of thing that would have a fairly significant selective effect, much the way that sort of like surviving childhood malaria might. And so we started to look at signatures of selection in our data. And so what we're plotting here is a measure of sort of more distant selection on the y-axis and more recent selection on the x-axis. You can see that in the tails of this, there's certainly a number of SNPs related to 1-carbon metabolism on this side. And in fact, a couple of these SNPs are actually the same SNPs that have been significantly associated in PRCL2. So that's sort of a positive thing. But interestingly, this is what we saw in Malawi. But in Jamaica, although the sort of overall distribution is the same, we don't see the same SNPs sort of coming out in the same way. I'm going to come back to that. So then we thought, well, that's the top SNPs. So what about 1-carbon metabolism SNPs in general when we sort of consider it, given that we do have the other 2 million and some SNPs that are on the H3Africa array? And so to sort of evaluate this in more detail, we sort of said, well, we're going to create a hypergeometric distribution by, say, starting with how many SNPs would one expect to see having a p-value less than 0.001? And so we have that number from the data set, and we then permute the case control status and do sort of 10,000 permutations of that so that we can develop a hypergeometric distribution. And then we can just ask for actually our observed data set. What is the, where does that fall? And that's where this red line is, showing that one carbon metabolism SNPs, the number of one carbon metabolism SNPs that are significantly associated at this cutoff is well out in the tails of that distribution. So you might say, well, of course, because you're sort of looking in a particular pathway and just comparing it against sort of randomness. But if we do that with a different pathway set of SNPs, so here is sphingomyelin, we've done it with ketones as well, we don't see the same sort of broad distribution. And if we use one of these other pathways as our background and do the permutation there, again, one carbon metabolism SNPs are still way out in the tails. And in fact, if we change the p-value thresholds, instead of 0.001, we sort of go all the way down to a more permissive cutoff of 0.01, we see that the resulting z-score still has one carbon metabolism SNPs way out in the tails of this distribution. So that was sort of encouraging that, this overall approach might be interesting. But what we did notice is that we still saw these differences between Malawi and Jamaica. And of course, we also saw this when we looked at the haplotypes that are associated in each of our top regions. So even in a region where there's very strong and similar linkages to equilibrium, the top haplotype that's associated is basically the mirror in one group versus the other group. And where we have less linkages to equilibrium, then it's even more of a mess. So we're not at anywhere close to a causal allele in any way. And what we're likely dealing with here is differences related to their genetic ancestry. So, so in one sense, we're fortunate in that we can see that all of these unprincipled components, they all sort of cluster together as African samples, but you see a very tight cluster of the Malawi samples in red, whereas the Jamaican samples are much more diversely spread over these first three principal components. And that sort of spoke to us to maybe think more about admixture in these individuals. So again, here, each column is a different individual, and the different colors represent sort of relative parental ancestries. So what you can see is that, looking at the ancestry of Jamaica, it's most closely related to the Yoruba, the West African in Nigeria, which is not unsurprising. And then there's some European ancestry as well, similar to the Ceph from from Utah. Whereas for Malawi, we see an ancestry that looks a lot more like Central Africa and southern parts of Africa as well. And so, when you think about the prevalence of kwashiorkor and edematous disease, which is much more common in sort of central and southern parts and gets less common the further north you come, you could imagine that there might be sort of a specific ancestry component that's carrying a susceptibility allele. And it might then, say, for instance, like this pink region here, might have different frequencies as you move. And that might explain some of the differences that we're seeing between Malawi and Jamaica. And so one way that we're trying to get around this is to think more about local ancestry. This is just an example from one individual from Jamaica, where you can sort of find this distribution. You can refine your association studies by knowing what the ancestry is at a particular locus and adjusting that way. And we hope that that will give us a sort of increased power to be able to tease apart these observations. So of course, this doesn't really necessarily answer the question of why you get one and not the other, although it likely contributes to it. And so we are, of course, interested in looking at a larger GWAS, for which we think we're probably going to need some larger numbers. And so, if anybody's interested in collaborating on that, we'd be very interested to hear from them. But I'm going to otherwise just stop there, just to sort of emphasize that there is this re-emerging role for one carbon metabolism, which we see somewhat bolstered by the genomics that we're doing. And that this integration of different omics can give you different and unique insights into some of these disorders. And then, as I said, we're working on this. We're sort of trying to push forward on all fronts that we can. And of course, as I mentioned, are very interested in sort of expanding the footprint here with willing collaborators. So I just want to end by acknowledging the many people who are involved in bringing this kind of work to fruition, and in making a shameless plug that in our new position at the NIH, we have postdoctoral positions available for anyone who's inclined towards sort of population genetics and genetic epidemiology. Feel free to contact me. And then with that, I am happy to take questions.
See, does anyone have any questions for Neil? Okay, well, I do. So if you're thinking about, you looked at the selection, and you showed that there was towards the end, that there's this kind of transect, a differing level of prevalence or incidence of disease. Did you look at selection in different populations to see if you're kind of seeing it some places and not in others?
Yeah, so I think that's what we were seeing a little bit. So we could see that, when you look in Malawi, we saw some things that were more out in the tails than the group in Jamaica, which might be consistent with that.
Well, I'm thinking about other African populations. If you look at databases and see what's going on there.
Yeah, so that's kind of on the to-do list, is working with the H3Africa group, which is work we've been doing with Ananyo, who's in the other room, to both look at the signatures of selection, but also the frequencies of these variants in the related populations.
Sorry about that. Yeah, because that, when I see those kinds of things, I always say, well, you've got different prevalence in different populations in Africa. It's very diverse. Maybe there's something that's driving selection in one population and not another, and then you kind of get this kind of very complicated, which is what the world is, right? It's
right. Really, yeah. I think that's kind of what we're learning, is that, especially when we're doing these kinds of studies in these populations, that you sort of have to be mindful of this very deep ancestral tree, and the fact that you might have very different drivers in different conditions. And so there's a lot more fine mapping that tends to be involved in this than maybe in a European or Western population.
Okay. Are there other questions? I don't see any, because I have other questions that I go, which are,
I have a question, Neil.
Yes.
Thank you very much. This was a very interesting presentation to see how translation research in complex disease situations, and I'm particularly amazed how the edematous severe acute malnutrition is called a complex condition, which indeed is for our infinite number of years as pediatricians. We have been all baffled about how complex it can be from various aspects, whether it's nutrition, psychosocial, genetic, microbiomes, microbolomics, and all those things. So my question was, what's the relation between the genetics you have seen, the CPG and the methylation and things, as compared to the microbiome, the gut flora? Is there a role or interaction there? And are you studying something to interact there and see if there is something?
Yeah, so I think that there is. So as you say, it's very complex. What we've observed is that the microbiota studies sort of show that there's a quantitative difference, that a shift that occurs. Some of Jeff Gordon's work had indicated that the shift that occurs, you get a shift away from the kind of bugs that produce some of the same substrates that are involved in involved in 1-carbon metabolism. So one way we're thinking about it is that you do get microbiota changes, but it's really the change in the metabolism of the microbiota. And that may actually sort of, both things feed into the metabolomic differences that are being observed. So we think there's sort of a synergy of things that are occurring on both sides. And so
a synergy and interaction somewhere.
Right. And so one of the things we're thinking about is, if you could supplement sort of 1-carbon metabolism, or you could change your microbiota back to something that would, also change the 1-carbon metabolism and availability, then maybe that's the kind of intervention that might sort of abrogate the progression of the disease.
Okay.
So I guess Jennifer had a question about the local ancestry analyses. So basically she asks, do we have a good examples of where this has helped? I think it's intuitive, but sort of, are there concrete examples? So we've actually done this. It's basically just an admixture type analysis. We've done it with alloimmunization in individuals with sickle cell disease, where we could see that there's an allele that is exclusively on an African ancestry background that doesn't occur in non-African populations. That sort of significantly increases your likelihood of developing alloimmunization, which is just sort of getting antibodies to red cells after a blood transfusion. And so what we see is that local ancestry analysis, like if we did the straight GWAS, it doesn't quite reach genome-wide significance. But if we do the contingency analyses, then we increase the power. And so now it's like very significant. And you're able to sort of see the association a lot more. Now that's sort of more recent admixture, right, between African and European. So, trying to now take it to sort of multi-way and sort of deeper ancestry is the kind of thing that we're working with other groups in order to sort of tweak exactly how that's done. But I think that that's the way that we have to sort of think about getting at the complexity when we have these kind of differences in prevalence.
Okay, I think we're going to have to end it now. Yeah, okay, well, thank you, Neil. I think that we're running a bit over, so I think we're going to have to move to the next talk. Thanks for a great talk. Really appreciate it. And it's interesting how I love when people can do really cool stuff with relatively small sample sizes. They ask the right questions.
Okay, thank you.
Thanks. Okay. And Kareem Manjai, my co-chair, will introduce the next speaker.
Yeah, so the next speaker is Professor Marceline Ngonu. Excuse me and forgive me for my pronunciations. Professor Juge holds a PhD in biochemistry since 2009 with a specialty in molecular virology and viral genomics. He is Associate Professor of Biochemistry and Molecular Biology at the University of Yaoundé in Cameroon. And she is a Fellow of the Cameroon Academy for Young Scientists. Marceline is an expert and a consultant in the field of human health research ethics, coordinator of ethics committee at the University of Yaoundé. She is a Fulbright scholar, alumni, and a visiting professor of Yale University in the United States. Her research on viral genomics and genetics has produced a number of articles in peer review and impact factors which are very high, such as the Lancet, the BMC Infectious Disease, the BMC Globalization and Health, and many journals. She has chapters in books and research. She's a, as you can see, she is a member of the review board of the BMC AIDS Research and Therapy, BMC Globalization Health and Academic Journals, and to date, she has trained at least 12 master's holders and currently supervises more master's degree students and co-supervises 8 PhD students. She's all these are in biochemistry and molecular biology and immunology, both at the University of Lyon and elsewhere. So, with this short biography, I would like us to welcome Professor Marceline Marceline Gounod to welcome her to give her presentation. Professor, please join us to impart some knowledge. We are looking forward to hear you.
Hello, everyone. I'm honored to be part of this 13th African Society of Human Genetics in Dar es Salaam, Tanzania. The research work I'm sharing today is entitled Construction of Hepatitis C Virus Infectious Molecular Clones from Cameroonian Isolates, an approach for vaccine development. My name is Marceline Gounod from the University of Yaoundé 1, and I'm a Fulbright visiting scholar from Yale University. The outline of this presentation is as follows. There will be a background, introduction, and then the problem statement in the rationale, the research objectives will be looked at, the HCV strains that circulate in Cameroon and the related clones, the replication of HCV in cell culture, and we'll conclude, give the selected references, and acknowledge some people. Hepatitis C virus infection is associated with increased morbidity and mortality worldwide. You all know that today there is no vaccine available for HCV infection. In Cameroon, HCV infection is characterized by a greater genetic diversity that could represent significant threats and challenges for successful treatment and response to potential HCV vaccine candidates. The laboratory-derived HCV infectious clones have been largely dependent on the Japanese fulminant hepatitis JMH1 strain. And this has offered limited opportunity for researchers to study the mechanisms of pathogenesis of that virus, as well as the reactivity of isolates to promising vaccine candidates. Researchers need representative isolates of each HCV genotype to be able to develop some robust cell culture systems that will facilitate further investigations. So the aim of our study was first of all to generate the HCV clones from Cameroonian isolates and to develop the cell culture systems using the isolates from Cameroon and from other parts of the world, Asia and America, to compare the different strains in cell culture for the various experiments to carry out. For the first objective, for the first research question, we identified HCV isolates, and these were transferred from Cameroon to Yale University in the United States for investigations that included molecular characterization of HCV, gene cloning, and cell culture. HCV capsid and NS5B genes were amplified by RT-PCR, nested and semi-nested PCR, respectively. PCR products were resolved in 1.5 agarose gel electrophoresis. These products were purified, cloned into a topo vector, and the generated clones were used to transform the chemically converted DH5 alpha cells. The plasmid DNA obtained from these experiments was used for DNA sequencing using the Sanger technology, chain termination sequencing. And the nucleotide sequences were analyzed using the Genius software. This allowed to generate the consensus sequences. And these consensus sequences were used for phylogenetic analysis through the NTDI BLAST. These experiments successfully analyzed HCV isolates from Cameroon were shown as belonging to genotype 1 that was predominant, followed by genotype 4. And the NTDI BLAST showed 77% similarity with the HCV subgenotype 1B, and also showed that the genotype 4 was belonging to the subgenotype 4A. And these subgenotypes are really virulent strains that require special care, including the appropriate direct-acting antivirals, such as the NS5B inhibitors. From the HCV replication in cell culture, it has been shown that some cell effectors play a key role in the replication of flaviviruses, as well as the hepatitis C virus. Some autophagy-related gene named ATD5 expression is upregulated, increasing autophagy, preventing caspase activation and apoptosis, and it also functions in the immune system, regulating innate and adaptive immune responses, including macrophage polarization, cytokine secretion, and antigen presentation. Another protein called LCE3 is known to form a stable association with the membrane of autophagogens. LCE3 antibodies are therefore useful biomarkers to detect autophagy. So it has been shown that ATD5 and LC3 knockout cells are autophagy proteins involved in the HCV replication. So the dissection of the individual steps of the HCV life cycle suggests that this protein specifically modulates the onset of translation of incoming HCV RNA and therefore the establishment of HCV replication. So if the cell effectors HCV5 and LC3 are used as to develop the cell culture system, it might play a key role in the replication of HCV. So isolate and RNA samples from the US and from Cameroon were used, including the positive control that was the wild type JC1 GLOC. These were were used to transfect the human hepatoma cell HU7.5. The experiments like the HCV infectivity assay, the anti-NS5A staining on samples were performed using these self-effectors so that we could determine which type points are necessary for the virus to be released in the medium. The medium tissue culture infection dose permeability was therefore determined for each sample and time point using the infectivity calculator created by Lindenberg. Medium tissue culture infection doses permeability were entered on a spreadsheet using the GraphPad software, and the growth curves plotted. The experimental conditions were 96 well plates format, and all samples were done in triplicates. The medium was DMEM supplemented with fecal cow serum. The cell seeding density was 6.3 per wall in 80 microliters. We performed the serial deletion, and the time points were 4, 12, 16, 40, 24, and 48 and 72 hours. And we use the positive control and also the negative control. From this experiment, as you can see, the growth curve. The HCV replicates very fast between 24 and 48 hours time points, so that the medium tissue culture infectious dose per milliliter reaches the maximum. But after 48 hours, it is observed that the curve it decreases, meaning that viruses start to die in the medium. So there is no more release of viruses, infectious viruses, or they are dying after the 72nd hour time point. So obviously this means that the appropriate time for this experiment, cell culture experiment, for the replication of this is determined. As take-home message, I would like to say that virulent HCV strains, subphenotypes 1B and 4A circulating Gabon, confirming the chronic infection noted in the country and requiring the use of NS5 inhibitors as DAAs. It is obvious that Cameroonian HCV strains, as other strains in the United States and Asia, replicate in cell culture. More experiments are needed with a larger sample size to hold the hypothesis. This includes also the generation of full length of more genomes, more representative strains, and the HCV RNA in vitro transcription for the translation experiments. But it must be known that the determination of the optimal time points for HCV replication is critical for vaccine development, preclinical drug testing, and basic virology research. And these optimal time points are from 24 to 48 hours time points. So these are the selected references. I would like to thank the J. William Fulbright Commission for the grants for us that allowed us to carry out these experiments, traveling to the US. The Yale University Wire Center for Molecular Medicine for the access to robust experiments, and the Lindenberg Laboratory for the collaboration and the facilities, all patients who accepted to participate in this study. Thank you for your kind attention.
Yeah, why don't we try that? Clayton, can you put me into the main stage? Hi. Marceline is having trouble getting online, so we'll take her questions after the next speaker. It's my pleasure to introduce Emil Chimusa. He is a PhD in computational biology and bioinformatics. He currently serves as associate professor at the University of Cape Town, where his expertise is in statistical genetics and population structure and genome-wide patterns of variation within and between species, where he's trying to address a series of fundamental questions related to biology, anthropology, and medicine, a very broad range. That's something I appreciate. He has extensive experience in designing and applying statistical methods to large genomics data sets in computational biology and statistical genetics. And his topic today will be dilemma in genomics risk prediction, African polygenic risk scores.
And sit here. Yeah. It's easier than to be here.
General ability and clinical utility. And I am not even sure, is he online yet? Okay, so he has a pre-recorded session. Okay, well, why don't we upload it now then? Okay.
Oh, yeah.
Risk predictions with a hypothesis that African polygenic risk score may enable generalizability and the clinical utility of it. So my name is Emil Chimusa. I'm associate professor at the University of Cape Town, and I have a great passion in human genetic variations in terms of drug response, treatment variations, and disease susceptibility. So today I'm going to actually give you a bit of overview on polygenic risk score, and I'll show you some benchmarking work that we have done and some results so far we got on some infection diseases and also general population for UK Biobank. So as you know, genetic variations reflect human history actually, and we know, all of us, we know that 100,000 years back in time, you know, humans migrate from Africa and move around the world. And that actually impact or contribute to the variation. And we all of us know different factors that actually contribute to variation that I've already say in terms of disease susceptibility, treatment and drug response. So those are actually also mutations that occur often and it's random, and natural selection, population bottleneck, admixture, and more other factors. So those factors are well done, but that led actually genetic has actually today huge diversity problem that need to be solved. Just imagine that if you have no information about Michael Jackson, no news at all, you never heard about him, and actually right now this gentleman appear in his room, and I have to ask you if Michael Jackson is African or European. So based on what we know about race by just the skin color, the type of the hair, you can use that to actually predict that, say, look, this gentleman is European, you know. So that may tend to be wrong actually in reality, you know. So now, if you got the DNA of Michael Jackson, now you use a kind of machine learning tools, you can be able today, we use that to predict actually the ancestry. And then you can tend actually to say he's an African American, where some part of his genome come from European ancestry and some part of his genome from African ancestry. So therefore, diversity is a critical for equitable translation of genetic studies. So we come from long path just to understand this kind of variations. So a lot of omics data initiative has been taking place since 1970. That was the era of DNA sequencing technology, starting by Sanger sequencing and then move forward. And I just highlight here for you just recently, since 2009, there's quite a lot of and rapidly advanced in generating data. A lot of consortium has been taking place, and many initiatives, UK Biobank, 100,000 genome from UK, 100,000 genome from Australia, from China, and also African genome variation project that's come to play. So all of this data has been generated. The question is still how many data we need actually to capture variations, and to understand this complexity of biological complexity, and also how many sophisticated machine learning we need to actually leverage this data that's already there to understand variation. Those are actually different questions that is still unsolved and is still actually a kind of a dilemma in this era. So a part of data initiative, there's actually a lot of approach has been generated, starting from 1911. That was the beginning of genetic linkage studies, and then move forward to 2005, beginning of GWAS era. 2008, microbiome study started, and till today. And the question is, what next today? What can be next approach to that can actually allow us to understand the variations, and that variation actually can have impact or application in a clinical setting. So far the progress has been done so far, but however, most of the study has been conducted in European context. As you can see from this picture, take this picture from my recent publications, that show 81% of genome-wide association studies has been conducted in European. You can see in the red over there. And only 3% in blue here in Africa, that's genome-wide association has been conducted. That is really show a huge disparity in the genomic studies, and that gap actually need to be filled. So in terms of data generation and in terms of appropriate method that can account for diversity of different populations. As we know, genome-wide association is studies based on the concept of common diseases and common variant. And it's also leveraging the linkage disequilibrium. And it's well known that the population undergone population bottleneck has a long range of haplotype or linkage disequilibrium than a population who are diverse. That is, I'm talking about European one, they gone a population bottleneck, and they have a long range of haplotype, and potentially GWAS has been generated and can be run properly to this kind of populations. So while African population have a high diversity and high admixture level, and that will require actually an appropriate GWAS approach to capture this kind of genetic characteristic. And in addition, African population are well known to actually undergo also selection pressure that can also some things to actually to capture in different methods of disease scoring statistics. So far, the question is still, even though the GWAS has been done in European context, European populations, but why there was a lot of method or tool of GWAS being actually implemented, as you can see on my right panel over here, since 2007 until today, last year. So there's more tool has been actually developed. So to actually capture the so-called stratification issue in GWAS and other potential issue that can eradicate the false positive and also boost the power of GWAS. So, but the question also still remains on those different tools. What can be the appropriate tool to be applied to diverse populations like African populations? So each of those tools has been benchmarked independently, but there was actually no way or no study actually benchmark those two in African populations. So we take a step forward to do so, whereby we actually simulated case control and with, of course, causal SNPs, that the data case control that mimic African structural populations, and also case control that actually mimic European populations, and also admixed population, where we simulate five-way highly admixed individuals in five-way fractions. So what we did actually, we also tried to see the rate of false positive in most of public tool, common use GWAS tool, as you can see on x-axis of this plot over here. So the first plot diagram here show three scenarios. That's mean on different sample size: 500 case control, 1,500 case, and 5,000 1,500 control. And also the reds line here is the plot of 2,500 case and 2,500 control. So those are actually different colors. So on y-axis, we try actually to plot how those tools actually can generate false positive. So that we have a baseline on the simulated causal SNPs, and we run GWAS on those different data based on different samples and on those each tool, and we actually compute the proportion of false positive. As you can see, even for African samples, those different to Emax, Gmax, GCTA, Plink, and so on, all those tools have always a certain proportion of false positive in African population, in European population, and also, of course, also in admixed populations. So then we see our observations across different populations, even including admixed simulated populations, that Emax that actually implement a kind of mixed model within a kinship matrix that's actually try to account for relatedness between individual within your a cohort, has a low rate of false positive. In addition, we try to understand how the replicability of GWAS conducted in European population in African populations. And we run meta-analysis using fixed effect model here, and also random effects, random models, and also binary effect model in European GWAS, simulated GWAS, and African GWAS. So our observation, we have seen that if the effect exists in both two populations, European and African populations, the appropriate approach to use should be fixed effects and has a low rate of false positive. The reason why to assess false positive is because there's a great potential of utilizing the result of GWAS in further downstream analysis in kind of like polygeny risk score, you know, where there's a quite expected application in a clinical and a diagnosis and a deep phenotyping, and also clinical trial that I'm going to actually talk shortly about. So there's a persistent challenge, although there's a many tools that's been actually developed, and I'll mention most of two of them that people tend to neglect. Is the first one is inefficiency of GWAS in distinguish between inflation from the common non-bias critical relatedness to the true signal of polygenicity. So when we do QC in GWAS, we tend to do as much as possible to reduce population stratification. By doing so, we are losing a lot of data that may actually explain polygenicity. The second one is the imperfection of asymptotic bias distribution of a mixed model or logistic regression that actually impose us to remove SNPs with low malofrequencies. We are losing a lot of those rare variants that can actually explain polygenicity, or can contribute to the risk predictions. And even though today there's excitement to use whole genome sequence to cover more region of the genome, yet when you conduct GWAS, we always remove rare variants. That is the challenge that still remains, and it still remains how to resolve this asymptotic bias in mixed models. And also the question is still how far so far current GWAS model can account for different type of interaction, like a host microbe interaction, gene regulation, and host genetic interaction, that's actually known to actually contribute to the host phenotype. So now, nevertheless, even those challenges still persist, actually, there is a way to leverage GWAS efforts into routine clinical use, and also we can use all those GWAS efforts to actually enable preventative or predictive medicine so far. And the common method is known to be polygenic risk score, which is actually the estimate of an individual genetic risk or reliability to a trait or a diseases, and that's been computed according to the genotype profile of an individual and also relevant causal SNPs from GWAS. So these formulas are actually where you have the effect of the causal SNPs and the individual genotype, and you try to aggregate all those small effects, and then you can actually estimate the risk or the genetic reliability of individual, and of course compare it against the rest of the individual within the populations. So today, PRS has taken a lot of attention from media, clinical applications, from the research, and also from the funder perspective. So it's actually important considerations, which is actually we have to bear in mind that causal variant effects are mostly shared across populations. And the way we are aggregating or sum up the small effects of individual within its genotype, the question is how you model these weights here, the beta, and this actually comes from GWAS summary statistics, and a different method has been generated to actually model this way people can account the differences in malofrequencies, the linkage disequilibrium, the model also family history, environment, and other factors. And that's how I'll describe in the next slide how multiple tool has been generated just to the way to model this kind of sum or this kind of weight within the genotype. So there's a quite, as I was saying so far, this PRS have actually potential to into the application at the routine clinical use, and to enable preventive and predictive medicine, and also with into the application of differential diagnosis screening, and also for deep phenotyping. Someone want to actually do phenotyping, you can use PRS actually to do to conduct a deep phenotyping, and also for clinical trial where you want actually to have a participant with high risk. You can actually apply polygenic risk within a bunch of individuals, estimate polygenic risk, and rank individual with a high potential risk based on particular diseases. So that is how PRS can be used. So far, there's, as I say, based on this weight, actually trigger a lot of research to generate more tools that can try to model malofrequencies on how many SNPs, causal SNPs need to be summed up to actually determine the risk of an individual. Those are different questions and those different equations have triggered also different tool developments. So far, there's quite a lot of tools so far has been developed since 1990 until last year. There's still more tool of polygyny risk score that's been developed to capture the way to actually model the weight. But also one of the things is those tools has been developed and been applied in European cohorts, as GWAS has been conducted extensively in European descent populations. So now here we try to benchmark those tools here in African populations known of high diversity. So we actually simulate case control at the first instant just to see how accurate those tools can produce a predictive power, a comparative predictive power in African populations. So we simulate case control, and also we simulate a true proportion of variant explained, which is actually over here, and run each PRS tool on that summary statistic that we simulated, that actually coming from case control African data. And we can see over here, most of those tools have a similar magnitude of predictive power or variant proportion of variant explained, and all of them, they are far away to reach the true simulated predicted power or proportion of variant explained. So that's means there's something is missing in current tool to actually improve the PRS predictive power in African populations. Here at this instant, we actually use African trend data and African target data summary statistics, and none of those two actually reach, a true predictive power. So we was thinking maybe there's some genetic characteristic that is missing in this tool, and we try to actually to evaluate the relationship or the impact of selection on effect size and on also allele frequencies, and to see if we capture selection on its impact on effect size and on also allele frequency, if that model can increase the predictive power. So this is actually what we got over here in this plot B. Under strong selections to a high trait value, one can actually expect a more U-shape, as we see over there, a U-shaped side frequency spectrum, and a high trait increase also allele frequency. However, the reverse is true as well. Under strong selection, there are a few causal variants since more have fixed, but a high fraction of novel rare variants are trait increasing, which result in the mean trait increase allele frequency being lower than 0.5 down on the plot that is there. So therefore, the message out of this simulation, where we try to understand the relationship of selection pressure and effect size and the spectrum of allele frequency, is that strong selection results in a weaker effect, and that can limit deviation from the optimal trait value, which may help to actually explain polygenicity. So from here, actually, we have actually been convinced that if we can model, when we are weighting the sum in a polygyny risk score, if we capture selection, the impact of selection in effect size. So in this, we can see over here that the predictive power of polygenic risk score can actually increase, particularly in African populations. So in doing so, we try also to figure out if someone use a European cohort as a training in a training models, and African population or other diverse population as a test model, how the accuracy, the behavior of PRS can be. So here we go. We try actually to model PRS by modeling additive effects of global ancestry and also local ancestry and the SNP effects, and also their interactions like SNP effects also with ancestry effects. That's how we try to model. Once we model that, we actually apply this model by implementing a less penalized regression, where we compare PRS predictive accuracy between European ancestry from UK Biobank as a trained models, and compare that to East Asians in a test as a test model, where we use kind of 8,636 sample from South Asians, 1,800 East Asians, and 6,983 African across 26 phenotypes. So this plot over here, as I was saying, European on x-axis, that was actually used. We used European as in training models, and South Asia as a test model. We can see that the correlations, the predictive power, the predictive accuracy is quite improved when you are using a trained cohort closely or genetically related to your cohort in a test models. And when we come to the African, we see there's a huge dispersion, across all the 26 phenotypes over here. So the take-home message over here is that the genetic risk predictions accuracy decay with increasing the genetic diversity. The more genetic diversity is between the train and the train or discovery cohort and the target data. We actually go further on to actually try to understand the prediction accuracy across multiple cohorts that is in the UK Biobank, so the continental ancestry. And as I was saying here, you can see the predictive power decay within the genetic diversity. The more the population is diverse, the more the predict accuracy of PRS decrease. So we look on a UK Biobank on only African ancestry, and we see that there is actually this kind of decay or differentiations of predictive power across within African populations, and is actually looks for us, it tends to us to be arbitrary, but also there's bias that's actually are not actually meaningful here. And we actually also see that the natural human evolution or the selection pressure can actually explain these differences of predictive accuracy among African population as there is here. So now, having all this information, we now use a cohort infection diseases, or HIV, in the Botswana populations. As you know from that the HIV have a high prevalence in Southern African population, as you can see over this worldwide map in the red here, high prevalence of HIV. And for us to actually perform polygenic risk score in these populations, we did actually use the whole genome sequences of 338 samples of HIV positive and HIV negative, type 1, and of which we have 226 HIV positive and 112 HIV negative, as you can see from this PCA plot case control over here. We perform a population genetics to understand the genetic variations in Botswana population by including other African populations. So clearly, our whole genome sequence of Botswana show that Botswana is actually sitting in the convex of quiescence and is West Bantu African populations, which actually makes sense for population genetic perspective. And we conduct GWIS on that. We found some SNPs to show up at level of genome-wide significant. And all of those SNPs actually, they've been nourished in the blood and also they are located in intergenetic regions of the genome. And also precisely they are in mtDNA and nDNA regions. So we take these summary statistics to actually perform progeny risk score, where we actually use our method where we actually account for ancestry, in additive model, the effect of global ancestry, the effect of local ancestry, and the effect of SNPs, causal SNPs, and also their interaction of ancestry and the effect of ancestry interaction with SNPs interactions, and also the model that we account for selections. So surprisingly, we got some results that show actually a very tight correlations between false positive rates and true positive rates. Bit of correlation that's seen that there's no actually the accuracy is actually promising here. And we compute the relative risk where we found 48% of the samples have a high polygeny risk score. And in absolute risk, we actually found 20.7% of the samples, regardless of HIV positive or HIV negative, to have high risk score. So this is actually some things that show our potential by accounting ancestry in Africa and selections in the method of polygeny risk score. So the take-home message here is that PRS polygeny risk score differ across African population, and that is arbitrary. But these biases are not meaningful. And the second thing is that the neutral human evolution or selection pressure can explain some of the differences in polygeny risk score. So at this point, there is actually a way forward where polygeny risk score can actually enable the predictive medicines, but this is still a question on how actually can be real, can be feasible, So our plan here is actually to leverage or to quantify the risk prediction predictable to a combination of host genetics, host microbiome, non-genetic element that is like a family history, even lifestyle, that's mean nutrition, and other environmental factors, and also include EHR, electronic health records, in the model of polygeny risk score to identify actually individual at a different level of risk for developing diseases. So I think by aggregating these informations, we might improve the accuracy. And also one of our observations is, even though we implement that, as we say that the PLS can differ across population, and that is arbitrary. So a way actually that to find a way to find like a consensus approach that can leverage multiple PRS tools to have a consensus risk that come from different approach, and also we need to actually improve education and training of a health professional in developing new risk communication tool. Also to consider randomized trials to directly evaluate the impact of polygenic risk score and the related individual health outcome. So that's it from me today, and I have to acknowledge colleagues and students and study participants and all research around the world who do research in GWAS and polygenic risk score, and I have to thank you. Thanks very much.
Thank you. Okay. Okay, can you hear me now? Oh, terrible.
What? 44, Marwan. Prof. Chimusa to join, yeah. Okay, Prof. Chimusa to join.
Can you hear me?
Yeah, I can hear you. Can you hear me too?
Yes, thank you, Prof. Chimusa, for joining. How are you?
I'm doing great, and yourself?
Good, good, yes. So, yeah, so we are waiting for any questions from the people who were listening to you on the presentation. So the floor is open to question Prof. Chimusa in relation to the dilemmas in genomics, risk prediction, the polygenic risk score studies from Africa, and the generalizability and clinical utility of this. Prof, maybe I can start. You alluded in your presentation about some diversity and a lot of heterogeneity of genetics. And in some of the reviews I read on genetic mapping, they find that in Africa is not homogeneous. And the more the different mixtures, especially in East Africa, then these genetics are everywhere. They come from Greece, they come from China, they come from India, from Asia, Arabia, and African. Whereas there is a little more homogeneity in the northern zone, like Morocco, Algeria, and things. What's your comment about this? And because one of the question is generalizability.
Yeah, so the generalizability issue, I think, is really a critical problem and very challenging. So as you can see, that's how people end up to decide to go for personalized medicine, where they can treat you based on genetic material. So there's a huge admixture in African populations, and at this point of the time, we are trying to resolve this problem by actually to try to predict the risk of a population by accounting for from some ancestry background. So the generalizability, it will not work 100% as per se, but as I say at the end of the work, at the end of my talk, I was saying that there will not be one approach for polygeny risk score that will be actually generalizable to all populations. But however, what we propose is actually to do a consensus approach where you use multiple tools, okay, and find a consensus risk predictions. This is just due because of this high diversity across population, particularly in East Africa, in North Africa, where there's a quite a lot of high level of admixture. So generalizability is really a big issue. Is a big issue. It can be solved now, but it's some things that we are projecting that if we can find a method, a polygeny risk score method that can take into account the genetic ancestry of a population and possibly some factors, as I illustrate in my talk, you know, environment factor, family history. And in that sense, now that too may tend to actually be generalizable.
Thank you very much. Any other questions?
Yeah.
Yes, I see, Scott, you want to ask something? There is a question here.
For you, Professor.
It says, did you track the PRS without effect sizes? That is an unweighted set of scores, as the effects are unpredictable amongst populations.
Yeah, so if I understand the question properly, is about if I did try to effect size. Yeah. So as you know, can you hear me over there? Okay, so you know, to compute the PRS, is a weight, okay, of a number of causal SNPs, okay, that you actually take into account when you multiply the effect size that comes from the genome-wide association summary multiplied by individual phenotype. So yes, that comes from the association studies, what we call GY summary statistics. But that is not sufficient, because that was the naive way to compute the polygeny score by using just the effect size that come from the association studies and multiply it by individual genotype. So we go further on ahead to actually compute, to design a model that take into account the ancestry, global ancestry, local ancestry, and also the effects, the interaction of local ancestry and the effect size in that model I described in the talk. So there's quite a lot of model, and definitely you need to account for the effect size of the association of a particular diseases with the genotype, and you have to multiply it by the genotype of individuals. You know, that was you know, a naive way for computing the PRS.
That's great. I have some more questions on the live question and answers. So, question Segun Fatumo, and she says, I understand PRS tools are not African data sets. I was really eager during your talk to see an African PRS tool developed in your group. Are you considering this?
Yeah, thanks very much, Segun. And I think Segun is a part of some of the ongoing work that we are waiting for the support for the funder. So yes, we actually do this kind of simulation in my group to access different current polygyny risk tool, and we actually try to implement a new approach to compute PRS. And we are still, as I say at the end of the talk, that right now we're going to try to account for family history, individual family history, for example, you know, and kind of medical health record as well in the model. So that is ongoing method that we are designing in the group. And I think we actually would like to apply this method in a multiple phenotype within African populations to see how the method actually perform within African population and African phenotype.
Okay. There's another question saying, how are you utilizing the PRS for deep phenotype?
Oh yeah, that's actually yeah. That is really good. You know, you can actually have data, right? You can have your DNA data, or maybe you know, candidate genes, or but at least sequence data of some individual, and now you want to choose actual extreme phenotype among those individuals. So you do the same things. You apply PRS. You can see in my last figure where we actually found, by applying PRS, we found some percentage of the sample have high risk on a particular disease. Those are the samples that you can consider for your deep phenotype. You just get them for the phenotype. So that is one of the examples. Another example is for clinical trial, where you actually want to actually select a group of sample that you use. If you have their genotype, okay, maybe it may be target sequences or whatever it is, you can apply PRS to actually select the most risky individual that you want to deal with them.
Right. There is another question or another comment from Segun Fatumo, and he or she says, "Great presentation, Emil. What kind of regulatory framework in using the PRS? Okay, what kind of regulatory framework should be considered before PRS can be implemented in in clinical settings in Africa?
Yeah, Segun, thanks very much. That is really important questions, and I can't give you answer now because that is ongoing work. I can't disclose it right now in public because that involves businesses inside. If Segun remember that we actually apply kind of funding, and that implicates some a company like Omics Data Solution, like other company in UK. So there's actually ongoing work that we is going on to actually fund policies on how to apply PRS in a clinical setting. So in my talk, I say, given the result that even we have with Africa, so is it possible to already start to apply PRS in a clinical setting? However, there is a that need actually a strict regulation and policies on that, and also the way to report the result, and the way to apply polygenic risk score, and the way to interpret it. Of course, there should be regulations, and as far I'm concerned on that, and the interest on that, I know Segun is also interested on that. So there's actually ongoing work in that regard. So there's a good example of regulation and the practice of polygenic risk score already in the UK on breast cancer and also on cardiovascular diseases. It works perfectly in European populations. We have that guideline and that regulation, and we would like to apply it in Africa to see how it works. You know, with a new method that we implemented, to follow the same guideline and maybe to work on that guideline to extend it more in actually African context.
Thank you very much. We have two more questions, and I think we will stop there. Number one, Tinashe says, great talk. Great talk, Emily. Thank you. Great talk. Most of the data presented is simulated. Have you tested these principles with real data in view of potential applications of simulated data?
Oh yeah. So that is if we actually in the talk we actually do synthetic data to assess different tools. That was a way for us to sense what's going on, what is the weakness, and so on. So where we simulate African populations and also European populations, we compare different GOS tools, and we apply just the synthetic data of African in multiple PRS, and we found that none of the current tool did reach actually a high level of accuracy or predictive power. So better now, we also apply those tools in a UK Biobank, European data, and also African data. And that was some of the plot there. And there is quite a lot of results that has been generated out of that work. But the conclusion was the PRS decay, as I say in my talk, the PRS decay with when the population gets high diverse, it decay, particularly when you are using European as a trend in the trend model. So we did quite a lot of scenario in that. And if you can see the last part of the talk, we actually applied that in HIV, and that's the data that we have. That was the whole genome sequence. And right now, I think we want to apply in many phenotype like malaria, cardiovasculars, some breast cancer, to see actually how that can work in African populations,
Thank you very much. Last but not the least, we have a comment by Gaston Mazando, who says, Emily, great presentation. Surely integrating different aspects of data sets will increase the accuracy. But how are you getting these different data sets? From the same sample or from the same cohort? How do you get different data sets from same samples or same cohort? How? Like same person, you get two different data sets, or? So this is a question from Gaston Mazandu. And unfortunately, that would be the last question in the interest of time.
Yeah, Gaston, I think thanks very much for the question. So we got different cohorts. As I say, for example, in UK Biobank, we did have this. It was the same cohort, but have a different 26 phenotype. Okay, European, but the same cohort with have information of 26 phenotype, same cohort, that have the same phenotype. And that's actually make UK Biobank very interesting data where we have the same sample, but that have many phenotype. That allow us to assess in the same cohort multiple phenotype. So however, in a HIV, we did have only one cohort that have only HIV, and that was the limitation of that. We could actually look on the same cohort that have many phenotype and test the PRS on the same cohort with different phenotype to see how the polygenic risk score predicts on the same cohort on different phenotype. That would be very interesting.
Thank you very much. I think we have reached to the end of an exciting parallel session this afternoon. Thank you very much, all the presenters, Professor Neil Hanchard, Professor Marceline Gonue, and Professor Emily Chimusa. Thank you very much.
It's my pleasure.
Have a good evening and a safe stay.
You too.
Bye. Thank you. Thank you, my co-chair.
Thank you.
William,
Every time I
and,
unmute my mic, I get incredible echo. So,
yeah. Bye.
bye.
Bye.